Hello guys, I am Dr. Mukul Mohindra. Specifically speaking, I am a sports surgeon, but my basic specialty that's orthopedics. And I'll be mentoring you for this subject if you subscribe for this unique platform, Unacademy. And yes, let me introduce you to what Unacademy is. Well, if your goal is NEET PG, then let me tell you, this is just the perfect place where you need to be. Because if you subscribe for this platform, you get to attend daily live classes. You attend them live means that you don't carry your doubt back home. Whatever doubt is there in your mind, you clear it there and then. And then even if you miss this class, the lectures stay as recorded videos with you. And then you can even download the instant notes once the class is over. And then you can further structure these classes the way you want. Test and discussion batch, regular course, uh, crash course, everything is allowed. And then you have an unlimited access with the same subscription price. So you just pay one time and you attend a single course or you attend multiple courses, everything is allowed. And of course, you can evaluate yourself also with a number of live tests and quizzes that are available on this platform. And all this comes at a price that is amazingly economical. Like, look at the subscription charges. Like, for example, even if you take the 6 months subscription, it just comes out to be like 3,000 per month. And then, if you further use this code Dr. Mukul-YT, you further add on a 10% discount to the subscription price. So, such a good bargain, I think. So I would be looking forward to have at least some of you on this platform Unacademy. But for the time being, I'll just try to show you the quality of teaching we have at Unacademy. So I'll just take you through exercise physiology, a bird's eye view. And that's my domain, so I thought why not discuss what you actually call as the nerve muscle physiology. We in sports medicine call it as the exercise physiology, synonymous. So I'll just give you a bird's eye view of how a muscle contraction is initiated and how a muscle contraction is controlled. All right. So let me first take you through the neurological control of a muscle. Now, this brain and spinal cord is basically what you call as the central nervous system. Branching from the central nervous system, you have the spinal nerves, and that's basically what forms the peripheral nervous system. So this is the spinal nerve branching from a spinal cord. Now, spinal nerve has a ventral and a dorsal root, and this dorsal root is basically sensory. So the sensory fibers, they basically travel via this dorsal root and synapse here in the spinal cord. So these are carrying information towards the spinal cord, the central nervous system. So they are basically labeled as afferent fibers. Now, depending upon what feedback is given by the afferent fibers, the spinal cord generates the efferent fibers. So efferent fibers will basically take the information away from the CNS. These are the efferents. And efferents basically get channeled into two pathways. The first pathway is the autonomic fibers. The second are the somatic fibers. What is the difference? Somatic fibers are basically those fibers which are under voluntary control, जो हम अपनी मर्जी से कंट्रोल कर सकते हैं. Like they basically go to the skeletal muscles. The skeletal muscles, we control their contraction. The autonomic fibers are basically those which are involved with involuntary control. So basically, they control the smooth muscles, muscles of gut, which are not under our control, and of course, they control the glands. And in order to control the glands, the smooth muscles, since autonomic system is not under our control, body controls them in two forms. there is a parasympathetic system and there is a sympathetic system. And the idea of having the two systems is very simple, very logical as well. If an activity is up-regulated by the parasympathetic system, it will be down-regulated by the sympathetic system. Or if an activity is down-regulated by the parasympathetic system, it will be up-regulated by the sympathetic system. So these systems counterbalance each other and ensure this involuntary control, positive, negative, to a smooth muscle or to a gland. These somatic fibers basically take care of the neural input, the skeletal muscle. So I hope you are clear. The central nervous system will branch into the peripheral nervous system. The peripheral nervous system would be the spinal nerves. They'll be having the sensory efferent fibers, and the efferents will divide into autonomic fibers, somatic fibers. The autonomic fibers will go to involuntary control systems, and the somatic fibers will be channeled to the skeletal muscles that are under our voluntary control. So let me just zoom into this area. How does muscle contraction get controlled by our voluntary efforts? तो ये थोड़ा सा मैं zoom in करके दिखाता हूँ कि how do this system works, the somatic system, the skeletal muscle system. Now please have a look again. This is the spinal nerve, the efferent fiber, going on to synapse with a skeletal muscle cell. Now inside this, the spinal nerve, the efferent fibers, you basically have the neurons. So this is the cell body, the axon. Now these neurons, they are actually arranged in clusters. And what basically happens that sometimes you want a neuron to fire strongly. So number of neurons will converge onto this neuron. A process that is called convergence. But at other times, you do not want this neuron to transmit the whole impulse. So this neuron will dissipate some of the impulse to another neuron in some other system or going to some other muscle. So this channel away of the impulse is a process called divergence. So in convergence, multiple neurons converge onto one neuron within this nerve, and in divergence, a neuron dissipates the impulse to some other neuron out of the system. This is all happening within this nerve. So within this nerve, this neuron and the axon, they are finally ending up in a skeletal muscle. Now please understand. This axon that is finally branching into multiple branches in the end. So I mean to say the axon branches, and the muscle at which it ends, so axon, its branches, and the muscle at which it ends, they collaborate to form something that you label as a motor unit. So this is what a complete motor unit is. The axon, its branches, and the skeletal muscle to which all those branches synapse. So please, towards the terminal side, before reaching the muscle, the axon will further branch into multiple small branches. So this one single branch would synapse with a single muscle cell, and this is what generates the neuromuscular junction. Yeah. So please understand that motor unit means the complete set, axon, its branches, and all its synapses. And within a motor unit, every synapse on a muscle cell is labeled as a neuromuscular junction. And yes, I've used the word muscle cell. So you can see the axon branching into multiple parts, going to a single muscle, but innervating multiple cells. So let me just zoom in as to what do I mean by a muscle cell. मैं एक बार ये भी नाट में dissect करके बताता हूँ. Now please see. This is a tendon and this is a muscle. The tendon is arising from a bone, and let us suppose this is biceps. Now within a muscle, you have multiple fascicles, and outside the muscle you have a layer called epimysium. अब इस epimysium के अंदर multiple fascicles हैं, surrounded by perimysium. So I'll take out one fascicle. This is the fascicle that is supposed to be surrounded by perimysium, Okay. Now, multiple fascicles make up a muscle, and within each fascicle, you have multiple small muscle fiber cells. So, a fascicle will be composed of multiple muscle fiber cells, and fascicle will be surrounded by perimysium. Now, I will take out a muscle fiber cell for you. So, ये fascicle में से मैंने एक muscle fiber cell निकाल दिया है. So, outside a muscle fiber cell, you will have endomysium. So outside a muscle epimysium, outside a fascicle perimysium, but if you take out this muscle cell, fiber cell, it is covered by endomysium. Now inside this endomysium of a muscle fiber cell, or simply inside a muscle fiber cell, you have multiple small filaments that are called myofibrils. So that will be my focus, a myofibril, because that is the basic unit where a muscle contraction is initiated by the somatic fibers. So what is the structure of a myofibril? Let me zoom into a myofibril now. Just see. This is the muscle, the fascicles, multiple fascicles. I've taken out a muscle cell out of a fascicle, and within this muscle cell, these are multiple myofibrils. Now, each myofibril has a light zone, a dark zone, a light zone. And within this light zone, you have a line that is called Z line, and this is basically what is labeled as a sarcomere, the basic contractile unit of a muscle. I'll just show you to recapitulate the first second prof when you used to read these things. So this is the electron microscopic picture of a myofibril, a light zone, a dark zone, a light zone. A light zone is called isotropic band, and this dark zone is called anisotropic band. Within a light zone, you have the Z lines, and within this dark zone, you have the H zones or the H lines. And what are these light zones and dark zones? Well, let me make you understand. These thin filaments are the filaments of a protein called actin, and these thick filaments lying in the center belong to a protein called myosin. So myosin filaments are lying in the center, and these actin filaments are lying on each side, held by the Z line. So between the two Z lines, this area is called a sarcomere. Now just understand, here you have just actin filaments, so it's a light zone and this Z line. Here you have multiple filaments interdigitating, so this will be a dark zone, anisotropic band, with a light line, H line. So that is the basic structure of a myofibril, and the basic unit of a myofibril, a sarcomere. Now, how does this sarcomere work to perform a muscle contraction? That is what I'm now going to explain to you. Just see. So this is what a sarcomere is. The actin filaments, the myosin filaments, the Z lines, and the sarcomere. Now, myosin filaments have heads called myosin heads. These myosin heads have to link up with the actin filament, and then the myosin heads pull the actin filaments towards each other. And when this happens, this sarcomere shortens. ये actin filaments पास में आएंगे, sarcomere छोटा हो जाएगा, and a muscle would contract. So what contraction basically needs is the myosin head articulating with the actin head, and then the myosin head pulling the actin ligaments towards each other. So how this happens? Let me zoom into this area also. So this is the myosin filament with the myosin head, and this is the detailed structure of the actin filament. So the actin filament is basically a helical filament, and actually it is composed of three proteins. There's a protein called troponin, and there's a protein called tropomyosin. So this protein tropomyosin basically covers the binding sites on actin. Myosin cannot bind with actin because tropomyosin is hiding the site. So what basically happens is that this magical ion calcium comes into a myofibril, attaches to this protein troponin, and then this tropomyosin moves away. And once the tropomyosin moves away, the binding site on actin is exposed, and the myosin head binds to the protein actin, and then the movement occurs of the myosin head, and then this sarcomere shortens and a muscle contraction is initiated by the help of this ion, calcium. I'll just show everything to you also, how the cross bridges form, how the contraction is initiated by this very nice animation. Just have a look at this animation. Okay. Basically what happens, so what I've shown over here is the myofibril, the sarcomeres. Okay, so I'll just zoom into this sarcomere to show you the whole story. Okay, yeah, so I'm switching on the animation. Yeah, I think, yeah, so just see, so this is the muscle fiber cell, and I've taken out a myofibril, and these are the sarcomeres, the Z lines, light zone, dark zone, light zone. So I zoomed into the sarcomere. So this is the myosin head articulating with actin, because the myosin binding site has been exposed by calcium. And now these heads will push the actin filaments closer to lead to contraction. That is how they push the actin together and lead to contraction. And once the energy ATP finishes, there occurs a relaxation. So that is how the sliding filament theory, the so-called sliding filament theory works. So basically, myosin has to attach to actin, and then myosin heads have to pull the actin filaments together, leading to shortening of the sarcomere and the muscle contraction. And yes, I must also talk about the source of calcium. I just told you that calcium binding to that protein called troponin is the initiator of the contraction. So how calcium initiates the contraction? Let me also brief you on that. So what I've shown is a muscle fiber cell. And as I've told you, a neuron would synapse on a muscle fiber cell, okay, to form the neuromuscular junction. Now I've just lifted up the cell membrane, the sarcolemma of a muscle fiber cell, and there are multiple myofibrils in this muscle fiber cell, multiple myofibrils. Now just see under this sarcolemma, there is something visible here. This is basically sarcoplasmic reticulum. So inside a muscle cell where you have multiple myofibrils, there's a rich network underlying the sarcolemma of something called a sarcoplasmic reticulum. This sarcoplasmic reticulum basically has two parts. One, the transverse tubules, and just by the side of the transverse tubules, you have dilatations that are called terminal systems. A transverse tubule in the center, and the dilatations on either side, they form a triad. So a triad means a transverse tubule and two terminal systems. This triad is the storehouse of calcium. तो सारा का सारा calcium इस triad के अंदर पड़ा है, transverse tubule and the terminal systems. But how would calcium be mobilized from this reticulum to the myofibril? This is what I'm going to explain to you. So please see. So what I have drawn over here, a neuron synapsing with a muscle fiber cell. तो ये न्यूरॉन मसल फाइबर सेल के ऊपर आके साइनेप्स हो गया है. सो व्हाट आई हैव ड्रॉन ओवर हियर इज ऑफ कोर्स द न्यूरोमस्कुलर जंक्शन. तो जैसे मैंने बताया था कि एक न्यूरॉन एक मसल फाइबर सेल पे जाता है, दैट इज कॉल्ड अ न्यूरोमस्कुलर जंक्शन. एंड लेट मी जस्ट क्लियर यू आउट, this area of the cell membrane that synapses with the neuron, this is called motor end plate. इसे मोटर एंड प्लेट के नाम से बुलाते हैं. जो सेल मेम्ब्रेन का एरिया होता है मसल सेल का, the area of the cell membrane of the muscle cells synapsing with the axon terminal, this is called motor end plate. Now the motor end plates or the neuromuscular junctions specifically are of two types. One are chemical and one are electrical. So basically, if neurotransmitter like acetylcholine is transmitting the impulse, it is a chemical neuromuscular junction. Or if a voltage gated channel, sodium channel, potassium channel is transmitting the impulse, it is an electrical neuromuscular junction. Now once an impulse reaches the motor end plate or the neuromuscular junction, depolarization occurs chemically or electrically along the sarcolemma or the cell membrane of the muscle fiber cell. Now this enters into the transverse tubule, and from transverse tubule, the impulse enters into the sarcoplasmic reticulum, the terminal cisterns having the calcium. And once the impulse reaches this area, the calcium is released into the cytoplasm of the muscle fiber cell, and this reaches the myofibril, and the myofibril undergoes contraction. That is the story. And let me just show you this area in a zoomed manner. So this is the transverse tubule, the cell membrane of the muscle fiber cell. So when the impulse goes into the transverse tubule, you have some receptors that are called DHP receptors, dihydropyridine receptors. So this impulse is generally electric impulse that switches on the dihydropyridine receptors in the transverse tubule, and these are coupled mechanically with something called ryanodine receptors that are present on these terminal systems, and they have a mechanical linkage. So the moment an impulse stimulates electrically the DHP receptors, it automatically stimulates the ryanodine receptors, and the calcium is released from these terminal systems into the cytoplasm to the myofibril, and the myofibril contracts by the sliding filament theory. So I hope you're clear with how calcium reaches a myofibril. I'll still clear your doubts by showing you a very nice animation how exactly this happens. So this is an axon terminal synapsing with motor end plate. So the impulse has gone down the transverse tubule, and this is the terminal systems of the sarcoplasmic reticulum. Now once the impulse will completely go through the transverse tubule, the DHP ryanodine coupling will occur, and calcium will be released into cytoplasm, and this will enter into the myofibril. And see the calcium entering into those myosin filaments, actin filaments. And just see now closely what happens. So the calcium has flown into the actin myosin complex, and this is that protein troponin, tropomyosin, hiding the binding site for myosin. येलो में बाइंडिंग साइट्स है मायोसिन की, ट्रोपोनिन ने हाइड कर रखी है, ट्रोपोमायोसिन ने हाइड कर रखी है. So now just see, calcium will come and attach to this protein troponin. ये calcium troponin पे attach हो गया, and see tropomyosin will move to one side. ये ट्रोपोमायोसिन एक साइड पे मूव कर जाएगी. So I hope you can see the binding site exposed. So actin पे जो myosin की binding site expose हो गई है, it has been exposed, and now the myosin head will bind with actin. Yeah. And now ATP, the energy source, will be broken, energy will be released, and this head will move, the actin filaments will move, sarcomere will shorten, and the muscle will contract. So that is the exact mechanism what happens inside a muscle fiber cell. So I hope you are clear that the efferent fibers, they go on to the somatic skeletal muscles, they form the neuromuscular junction. Every axon terminal branch goes on to single muscle cell. Inside the cell you have the myofibrils, and myofibrils have multiple sarcomeres. Calcium will go into the sarcomere from the sarcoplasmic reticulum, initiate the contraction by coupling the myosin head with the actin filament, and the myosin head will move by using ATP. ATP. So source of energy is ATP that is present inside cytoplasm of a muscle cell. So let me also brief you. ये ATP कहाँ से आती है? What is the source of ATP inside a muscle? Well, let me tell you. If you have to take a muscle contraction that is going to last very less than a minute, like you want to sprint 100-meter भागना है. So now the source of ATP is a compound called creatine phosphate. This is basically present in the cytoplasm of a muscle cell. This phosphate can be broken down from creatine. It can be given to ADP, and ADP changes to ATP, and this ATP leads to the power stroke. So this is the fuel for a short duration contraction, creatine phosphate. If the contraction has to go a little beyond that, up to 20 minutes, now the fuel becomes glucose, and you already know that this glucose basically lies in the cytoplasm of the cell, and from glucose ATP can be generated in the cytoplasm by a process called glycolysis. So glucose will be the source of energy, ATP, for a contraction between 1 to 20 minutes, and both of these, they are anaerobic processes. This delivery of phosphate from creatine, anaerobic. ATP generation by glycolysis, anaerobic, oxygen is not required. So when contraction has to go beyond 20 minutes, oxygen becomes a necessity. So now the energy source of ATP is actually the mitochondria inside a cytoplasm, where you have this Krebs cycle running, where fats are broken down by turning them into acetyl coenzyme. So this provides ATP in a longer duration, and maximum ATP is generated by Krebs cycle far more than glycolysis. So longer contractions can ensue. The idea of knowing the systems is कि तुम decide कर पाओगे कि किस केस में किस system को strong करना है. Like you want to train a runner, he needs burst of energy, so creatine supplements need to be given because contractions are going to last for long. You want to train a marathon runner, he needs more of fats because he needs longer contraction, so you need to supply fats, and you need to train him for activities that go beyond 20 minutes. So that is how we decide which athlete needs which kind of supplements depending upon the amount of activity is going to carry. So that is how ATP triggers a muscle contraction, and that is how ATP is generated within a muscle fiber. And yes, once ATP brings about a muscle contraction, how does a muscle decide what should be the force of contraction? Should it contract? Should it not contract? So what is that feedback mechanism? Let me also explain that to you. Now within the belly of a muscle, you have muscle spindles, and along the tendons, you have something called a Golgi tendon organ. The basic purpose of muscle spindle is to sense the length of the muscle, and the basic purpose of Golgi tendon organ is to sense the tension in the muscle. Now ATP is broken down, muscle has contracted, and you're lifting a heavy weight. This heavy weight will push your hand down. There will be increase in muscle fiber length, and now the muscle spindle by these afferent sensory and efferent pathways would communicate with the central nervous system to decide how much should the muscle contract to lift the load to the desired amount. And yes, sometimes the load is too heavy to lift. You might end up rupturing the muscle. So now the Golgi tendon organ senses the tension in the muscle, and when the tension is too much, via the afferent pathway it tells the central nervous system, and via the efferent somatic fibers the muscle contraction is inhibited because of excessive tension. So that is how a contraction is stimulated and stopped. The length that you want, that tells you how much a muscle can contract, but excessive tension would inhibit it. So that is the simple funda of deciding the amount of contraction. The muscle spindles in the muscle belly sensing the length, then responding. Golgi tendon organs within the tendons sensing the tension in the muscle, the stress in the muscle, and then responding. And how do I just remember? बड़ा आसान तरीका है. T tendon, T tension. So Golgi tendon organs basically sense the tension. They respond to tension, and response to length will be by the muscle spindles that will be present in the belly. Right? So that is how the amount of contraction is decided. How much ATP needs to be broken down? That is the question. So I hope you are clear with the whole picture. सारी चीज समझ में आ गई है कि somatic fibers spinal nerve में जाते हैं. They eventually reach a skeletal muscle that is under our control. How much a muscle can contract? The spindles and the Golgi tendon organs tell in a simultaneous manner. The tendon would sense the tension, the spindle would sense the length of the muscle. And yes, if a contraction is to be initiated, the spindle goes ahead, initiate kar lo, contract, it says to the muscle. The muscle contracts by the sliding filament theory. For that, the neuromuscular junction exists on a muscle fiber cell. In the cell, you have a myofibril, and the sarcomeres in the myofibril are the basic units of contraction. They have actin and myosin filaments. Calcium binds to troponin, and then myosin connects with actin, and the actin filaments are pulled closer, and a contraction occurs. Okay, so calcium comes into the myofibril from the sarcoplasmic reticulum by that DHP ryanodine receptor complex coupling, and I hope that you can clear that. तो ये सारी चीजें समझ में आ गई हैं. It's time to end the lecture with a rapid fire MCQ round. Let us see what all MCQs we have learned in this short session. Okay, the basic contractile unit of muscle contraction, sarcomere, because it is the sarcomere that contracts and that relaxes, and a muscle contraction occurs. So I don't think any more proof is needed. It is sarcomere that is the basic unit behind a muscle contraction, because it is the sarcomere that has the actin and the myosin filaments and the cross bridges and that sliding filament theory. calcium will initiate muscle contraction by binding to which muscle protein? So you very nicely noted, calcium binds to this protein, troponin. But here there are two choices, troponin C, troponin T. So let me tell you, troponin has two ends. The C end will bind to calcium and this T end will bind to actin. So T end binds to actin, the C end binds to calcium. So this will be the answer. So calcium will bind with the C subunit of troponin. AIIMS question. AIIMS 2020, the exam that has just gone by a couple of days back. The dihydropyridine receptors couple up with the ryanodine receptors. How? So I told you that this will be an electrical impulse and the DHP receptors would switch on the ryanodine receptors by a mechanical method, because this is a mechanical linkage and this is an electrical linkage. So that was the answer to this question. Tension in a contracting muscle will be sensed by T for tension, T for Golgi tendon organ. We've already covered it. And one last question, again, AIIMS 2020 question. So a lot of questions come from exercise physiology. A single neuron receives 50 inputs. So एक neuron पे multiple inputs आ गई हैं. I told you this is convergence. And when from a neuron impulse goes away, that is divergence. So here single neuron receives multiple inputs. So this will be convergence. And yes, I must also tell you what is summation. Please understand. Summation is basically at the neuromuscular junction. Just understand that if this single neuron fires again, contraction will become stronger. This is called temporal summation. But please understand, sometimes the impulse would make all the neuromuscular junctions fire together. All the neurons fire together. This is called spatial summation. So when all neurons fire together, a muscle can contract hard. Or when a single neuron fires so many times, even then a muscle can contract hard. Single neuron does it, temporal summation. Multiple neuromuscular junctions fire together, spatial summation. So I hope you're clear with all the basic concepts, because once you're clear with the basics, you can actually tackle any pattern of the examination. So that's all from my side. I hope you enjoyed the lecture. Many more free live classes are there on Unacademy. Watch them, or preferably subscribe for the platform to watch the plus courses and the batches, so that you just chill when others are in tension to see a tough paper. Please don't forget to use my referral code if you're subscribing for Unacademy to get you 10% off. Thank you.